Alright, so let's move on. So the third video is basically more or less more about human cloning, and we're gonna have a look at some of the timeline. But let's look at the basic first. What is the correlation between human cloning and genetic modification? So we know that everything starts with a gene, and there are lots of genes in our genomes. So we know that genes is the basic cell of life. It contains your hereditary material, which is your DNA. So it could create protein, and they are mostly found in chromosomes, and of course are located in the nuclei of your body cells. So this is your nucleus, okay, with the chromosomes, and the chromosomes contain your gene, which later we converted into protein. So to make an embryo, there are three ways or three approaches. So first, which we know, which a lot of people normally know, okay, is through sexual reproduction, whereby most of the time a child gets half gene from mother and another half from the father. Kind of like if you remember from last chapter in transgenic animal on the homologous and non-homologous recombination. So second approach is by asexual reproduction. So we are animals, we humans, but we don't go through asexual reproduction. Only plant goes through that. But by asexual reproduction, you could still create a child. You could still create a zygote where a child could get all genes from single individual's body cell or from both parents. So in vitro fertilization or IVF is basically one form of asexual reproduction, whereby the egg and the sperm is basically fused or fertilized outside of the human body. So apart from that, you could actually also create a baby by a single individual's body cells, but you just still need an egg to more or less let the fertilization occurs later on to produce embryo. Now, the third method is by parthenogenesis. So this is a type of cooling, but I wouldn't say the downside, but the restriction is that the child only have a single nuclear donor. Can be either from a man or a woman, doesn't really matter. And the good thing about parthenogenesis is that you could actually determine the sex of the baby beforehand. So you just need a polar body from either the man or a woman, and some, of course, egg nucleus. Fuse them together by electrofusion, and of course, again, with many cell division, zygote is produced, and then you could get a baby via surrogacy. So, what is parthenogenesis, and basically, how could it be used, okay, to make uniparental embryo? So, this we know, okay, what happens through natural mating and also IVF. But through parthenogenesis, okay, what happens that, okay, from the maternal, there is only one side, okay, either the man or a woman, okay, just have to choose. So it could be done either by artificial stimulation, okay, so these are some of the methods, by inhibition of MPF cyclin B, okay, and also by similar method, okay, such as inhibition of MPF is by blockade of second polar body extrusion by cycloclazine B. So you get a parthenode, which means that a uniparental zygote with two maternal genomes. So this is how parthenogenesis or parthenode baby is being determined in terms of its sex. So the sex determination system is basically by the ZW equals to, you get either double Z or double W or one of each, which is ZW. So with us human, we basically go through XY or X0, which means that XX. Okay, so for the female. But over here, double Z is for the male, ZW is for the female. If you get WW, what will happen? We'll get to that. Alright, so let's take a look again over here. What happens to when you get this, right? So we know that again, when you get a ZW, it comes from a female. So upon analysis, okay, so it were to divide. So if this, okay, you get another second polar body, and your baby will be ZX chromosome double Z, which means that it's going to be male. So over here, if the same division occurs, you get double W. What will happen is that this will die. It will not develop into any baby. Okay? So that means that, okay, if you were to get this, that means that nothing, no babies will occur, or no babies, okay, you won't get any baby, so it will die. The zygote or the egg, okay, will die off later on. Alright, so in terms of the stem cells, So we know that stem cells has many uses. So stem cells has been used to more or less in terms of therapeutic purposes, especially for gene therapy. So parthenogenesis is actually one of the method, okay, used to produce stem cells. So this is how, okay, it's actually being used. Right? So from this I got, you can just read on about it over here. Okay, but focus on this diagram over here, production of stem cells. So you can either get the zygote from natural or from clonal, such as parthenogenesis. You get the developing embryos from the blastocyst. Okay, you can just get the embryonic stem cells or the basically the inner cell mass. Basically, you could just harvest it and well grow it in the lab in vitro, and more or less induce it to become different types of cells, such as bone tissue or muscle tissue or nerve tissue for therapeutic purposes and so forth. Now, let's have a look, since we are on human cloning, the distinction, okay, between reproductive cloning and therapeutic cloning, because we have seen of the three differences early on, but we're going to focus only on these two. So with reproductive cloning, it means that it produces clonal embryo to be implanted into a womb. But for therapeutic cloning, and I think you would hopefully get it from the previous slides, it is, or the purpose is to produce clonal embryo used to generate stem cells for therapeutic purposes, for medical purposes. So the later, okay, is basically for tissue or organ creation. Now with human genetic engineering, basically you could change genes, okay, in a living human cells, and we know that with the discovery of CRISPR that is made even easier. So it's just a mean to manipulate the genes such as this. Now let's have a look at the differences, okay, between somatic genetic engineering and also germline genetic engineering. Okay, so the keyword here is one is somatic, one is germline. So if you understand the term somatic or soma cells, okay, versus germline, it means that, again, somatic GE target organ and tissue specific genes without affecting the human reproductive genes, which means that whatever manipulation that you did, okay, via somatic GE, usually it will not be passed on to the next generation. It will only affect the individuals taking place. But for germline, because it involves germline, it targets genes in the eggs, sperm, or very early embryos, those that is involved in reproduction. So of course it would affect every cell in the body and will have a very likelihood or high chance to be passed on to the next generation. So this is basically how the germline engineering combine the use of stem cells and embryo cloning. So you can get lots of things from the egg to be used either to produce stem cells or to generate or to clone another baby, parthenogenesis. Now, let's take a look at one of the approaches being used commonly, and well, the method is being improved, but this is basically the basis of the method: pre-implantation genetic diagnosis and selection, the PDS. So couples that have a child carrying their genetics but does not carry the genetic diseases, which means that, for example, you know that you have thalassemia, you are a carrier, so your child will likely have the thalassemia. So you could predetermine that. So basically, how the process is done is by selecting healthy embryo for implantation, and it works as in germline technology plus an IVF procedure, whereby only healthy embryo is selected and basically be implanted or returned back to the womb of the woman, so that when she gave birth, okay, the baby is healthy without the disease affecting either one of the parent. So it could be used to select for not just for healthy babies, but for cosmetic, behavior, and also non-disease trait as well. So this is how they do it, okay, in the lab. They test the good zygote first, okay, and then what will happen to the ones that is not chosen? Usually it will just be destroyed. So this is GE, natural selection, but a bit unnatural. So now let's have a look at the human cloning timeline before we proceed with the fourth video, which is actually quite, I would say it's quite heavy in the beginning, okay, but then it's still just the basic of human cloning. Alright, so in the timeline, okay, so I just put it here, this is from start of Dolly, because, well, even though it's nothing to do with human, but it's part of what it is, an animal, so we are an animal, so that is basically just the benchmark of human cloning. So what happens again is that, again, this is from the same year, again, so they probably try to propose a legislation to make sure that the process or the creation is not being seen or being more or less condemned as something which is not good or defying human nature and whatnot. So they are trying again to get more or less make it kind of like approved by the ethical committee. So in 1998, again, it proceeds. So lots more countries signed the treaty, okay, to make sure that even though this cloning technique again could be used to clone animals, but they actually do not agree for it to be used to clone a human. So basically, there are lots of countries over here. I'm not sure. I think in the actual PowerPoint you could click this, but because this is probably from the PDF, probably you couldn't from your notes. Alright, so same year, right, the Food and Drug Administration, the FDA, announces its authority to regulate human cloning, because when Dolly has been created, people thought that, wow, an animal, a large animal, larger than human, could be cloned. Why not human? So the idea comes from a lot of people, not just scientists, politicians and whatnot. So they do not want this to be manipulated. Hence, that's why they are trying to make sure that the process could be used to clone animal, but not on a human. Okay? So from then on, again, this is from Dolly. The creation of Dolly. Okay, so there are lots of animals that has been cloned ever since, as you can see. So these are some of the sites related to the clone animal. So if you were to go through this, you can see that before Dolly, some other animals has been cloned. Okay, so after Dolly, we see that okay, the goat has been cloned, called Mira. Cows, mice, human stem cells, not human, just stem cell. Tasmanian tiger, okay? Xena the pig, okay? Pigs, Indian bison. This is kind of a bull, okay, a large bull. So human embryos in 2001 has been cloned. We get wild sheep, we get cat, calf, African wildcat, deer, mule, horse again, and a lot of other animals. So from this list, can you see something which catch your eye or probably pique your interest? Have a look at it for about a few seconds, because I'm just going to try to pique your interest or see whether you are curious or not, or whether you know which one of the animals in here are actually extinct. Did you see? Did you notice? If you did, good on you. If not, then over here, woolly mammoth. It has been extinct for some time. Now imagine, woolly mammoth. It looks kind of like an elephant. It's going to be very big. If it is to be cloned and let roamed in the world, what will happen? Just imagine that. Alright, so I think I'll stop here for the third video. So we just move on to the fourth and the last video for this week.